Okay, it's not my view that I'm going to highlight. I'm going to present what we will be doing this afternoon, because all of you, all of us, because I'm a scientist also, I do phage display for a living. We use phage, some trying to understand them, some using them as tools. But the ultimate tool for the sake of humanity might be that it offers an alternative to the ever-growing problem of bacterial resistance. So phage might be the way out for a conundrum. And this is what this session is about. Everyone somehow feels that. But how to make it happen, how to do it? It seems obvious, but it's quite complex, and I want to introduce for a few minutes the challenge that lies ahead. So, as I said, it's a vision thing, because actually, the key aspect of bacteriophage, and you know that as well as I do, they're co-evolutive with their bacteria. They're bacterial killers, but if the bacteria escape its killer phage, well, the phage in a few hours, in a few days, will catch it up and control it in the wild. So that makes it the only sustainable approach, because you will never run after the bacteria, you will run on the site of the bacteria in terms of bacterial resistance. And this is our biggest problem. Such things make the news regularly in every country on the planet. But what's interesting is not the title, it is more here. We confronted, in dealing with the bacterial world, between a dynamic position, which is the germ mutate faster than the chemist can generate new molecule, if they want to. They used to want, but we have another problem these days. Such molecules do not bring enough money, so the pharmaceutical industry has qualms about devising new one, because it's a 10-year shot, 12-year-long project, $500 million or $1 billion, for a lifespan, effective lifespan of the medication of 2, 3, 5 years. Then it's worthless if it starts to be confronted with a lot of resistance. And so the IP, and this is an idea you won't hear elsewhere, but the intellectual property right can use a business model that contributes to the multidrug resistance. Because once a molecule comes on the market, let's say penicillin, just to be non-committal, after 5 years you see that penicillin is not functioning as well as it should. And it doesn't bring money because you reserve it to the extreme case. Then what you do as an investor or as an industry, you want to enlarge the clientele. So you want to overprescribe. And so you bring molecules that are initially reserved for the emergency room or for the clinic, and you bring them to the community. And all the medical officers or doctors around here know that we've been for the last 30 years under constant pressure to overprescribe. And there you have a big contributor to the multidrug resistance phenomenon. So the fact that you have to recoup your cost on 5 years for something that's protected 20 years. So essentially we have a mismatch between the intellectual protection, which is 20 years worth, with the actual efficacy, effectiveness worth of an antibiotic molecule. But that's another discussion. So what we will do this afternoon is to show some of these, that the interactions are dynamic with the phage and the bacteria, and that, like on the red curve, you can always catch up the bacteria genetically and bring them back to control while disposing of them. Some of us will discuss about the fact that when you have bacteria in a lab, you can have them co-evolve in the test tube. And I won't present these data because they're not mine and they're from Angus Bucklin's lab, but they're fantastic. Let's say that this is time of generation, and these are condition and type of bacteria, or a relation between bacteria, coupled bacteria and phage. In gray, it's resistance, and in white, it's they gotten back, the phage got the bacteria back under the control. This time scale can vary from 6 hours up into a few days or a week. So this is quite fast in terms of coevolution and the hide-and-seek game that the phage and the bacteria play together. What are the problems we're going to address or to solve this, to make phage therapy a viable alternative and a new paradigm for antibiotherapy? What are the problems? First, we have a regulatory problem because, and we had this discussion, we went to EMA, and some of the speakers this afternoon will talk and explain that in detail to you. The phage are not legal entity because they're not a vector, They're not GMO, they're not genetically modified organisms, but they're not pharmaceuticals because they're not stable. The composition will vary from year to year, or eventually from batch to batch, because you adapt the therapy to probably your problem, your bacterial infection. So essentially, the only comparison that could be valid is the vaccine, because with the flu vaccine, you have to change it once a year. It will be like the vaccine, but changing on a faster time scale and locally, because the flu vaccine is valid for the planet. The phage will be valid for a country of a portion of a continent for a few times. So basically, there is no model to put a regulatory frame around this. And the people are willing to help, but we have to start from scratch because it's not an advanced therapy, etc. We had this discussion, and it will be explained to you later. Then there are the societal barrier, because when I had the honor to present the first West-organized controlled clinical trial in the Western Hemisphere, we needed 1 year to get the bioethical approval. And I had to confront colleagues in good faith, very intelligent geneticists, telling me, but Martin, you want to give virus to fight bacteria. I said, they're not virus. In 500 million years, they didn't catch their way into our cell. They're harmless as far as genetic is concerned, if we use the lytic only. They said, yeah, but blah, blah, blah. So even doctors, even scientists were not convinced that this was a viable and safe alternative. And I finally convinced them, but if you don't allow it to happen, I will do it anyway because I know it's safe. So they say, okay, you got the clearance. And then I add up. And the cold cream of your wives, by the way, contain billions of phage per ml. So psychology is also an issue. Then you have the intellectual property framework that I already highlighted to you, the business model, whose time scale mismatch the efficacy of the drug. But it's not an attack on the pharma. We need the pharmaceutical industry. Nothing will get done without them. It's just that we have to negotiate and to inform them of new possibilities, novel way to think. And maybe it's worth, or it will be a time to open new thinking about the IP protection just for antibiotherapy. Because protecting a molecule 20 years when we know that it won't be effective 20 years is useless. So give and take, there's room for negotiation. And maybe why not get informed and see what happens in the software sectors, open source things that might help unlock the situation from the industrial standpoint. Then, of course, some of the science is missing. We all have heard for the last 20 years, oh good, the phage can work. But we don't know the studies. We don't know where they do not work. So we need to do quality control studies. We need to do typing of phage that we will reserve for human use, because not every phage is even powerful, etc. All this basic work has to be, validation work has to be done with the same strain. So there is coordination at the international level, optionally. And so intimately, we need a phage bank or phage banks. That's also part of the problem. And all these problems are linked. And to think that every one of us focusing on our single issue, because if I'm a molecular geneticist, I want to look at evolution. If I am a phage display person, I want to screen for interaction or for ligand. If I am a metabolism specialist, I'm studying metabolism. And we think that the field advances without us. So we need to pull together, because we need to solve those aspects at the same time in parallel. You cannot slice a project and say, we first solve this and then solve that. We have to work on the political level, on the legal level, on the science level, in parallel. There is no way out. So we have several ways of doing this. Either prêt-à-porter, that would be the industrial way. You produce phage that you could patent or repertoire, eventually for disease, or for a niche, or you can do it in special medicalized center in which you would get, for multi-resistance germs, the germ itself, co-culture, and make a batch for this patient. And in between, all is possible also. So here is a list of all the problems we have to tackle to really fully exploit the potential of phage therapy. And now you understand what this session will be about, because it looks like a funky program, but in the end, we will have speaker, the last three speakers of the afternoon, speaking and making the case for the paradigm shift. They will have clinical aspect. We will have data about coevolution. Then some of us will talk about the legal aspect. Some of us will talk about the proprietary aspect. There's still something missing, but we're only starting. In fact, it's already 2 years, we're busy. And I thank you for this for your time and interest this afternoon. It'll be a very interesting session because at the end, we will try to have a good panel to try to merge point of views and forge a way ahead. I thank you, and I give the floor back to my colleague, which will introduce the speakers. Isabelle.